Good morning. Oh, I didn't boot my camera up properly. Whoops. Fix that. And if you haven't done so already, please let me know you are present in the chat. Okay. So before we get started, please let me know if you have any questions from yesterday's content. So as a quick reminder, we covered a lot of ground. We talked about protists and the three types: plant-like, protozoans, which are animal-like, and fungi-like protists. We did a quick little summary of fungi: chitin cell walls, heterotrophic. Other than that, they look a lot like plants. And then we finished it off by doing a brief overview of plants, pteridophytes, bryophytes, gymnosperms, and angiosperms. We talked about the idea of alternation of generation, cellulose, cell walls, sessile, all multicellular. Chlorophyll as their photopigment. Well, I should say primarily chlorophyll is their photopigment. There are actually other photopigments in plants, but it's primarily chlorophyll. Any questions from the textbook questions? Stuff you were looking up for the notes? Nada. Okay. If you think of something, don't be afraid to throw it in the chat, and I can always circle back. So today, believe it or not, is the last day of Unit 1. Crazy. So that's typically the units will be about 5 days long. I think one of them's 6 days and one of them's 4 days. So I mean, it's in the neighborhood of 5 days long, which, according to the Ministry of Education, should represent approximately 20 days of class, which is shocking. We're supposed to do approximately 4 days of class every day. That's well, let's just say we're doing our best. We're doing our best. I've definitely had to modify things. That's very onerous of an expectation on students to do 5 days worth of content every day, 4 or 5 days of content. So it doesn't matter even if you have all the hours available, which I guess we technically do. That's not how the human brain works. You can't just squeeze all of that down into, a day and then expect students to remember things. And like memory takes time. You need recall over multiple days in order to put stuff into your long-term memory effectively. So anyway, we've basically taken the level of detail and knowledge from this course and kind of moved it, like decreased it a little bit, made it a little less deep. Not that we've completely eliminated everything. That's not fair to say either. I mean, I've kept a ton of stuff. Obviously, we're covering a lot of content, but I have removed a little bit of the depth of it in order to A, fit this timeline, and B, keep it reasonable for you as something that you could, actually place into your memory during this like advanced timeline. So this is the last day of the first unit. That's where I was going with this. This is the last day of the first unit. So what we're going to do today is finish off with animals. Then I'm going to have you guys complete a summary sheet for the various kingdoms. Okay. I have a completed version of it that you can have a look at if you want to double check your version. But it's essentially a table that gives you the basics for each kingdom. What are your takeaways that you should be able to identify for each kingdom? That'll help as a study aid. It's basically like you have to go through and review your notes in order to fill it in. And it'll also be a handy reference for when you are doing your quiz. So that's the first thing that we're going to do today. We're going to finish off animals, which won't take too long. Again, we're not getting into super deep depth in each of these kingdoms. And then we're going to complete that summary sheet. And then after that, I'm going to give you the rest of your time today to work on your species spotlight assignment. I just realized that nothing that I'm pulling up here is actually on your screen. Let me try that again. Sorry, I'm referencing the stuff that I'm talking about here on Brightspace, but I just don't have it on my screen, on your screen. Okay, here we go. So, as mentioned here, we're going to cover animals. Animals is actually on day 4. Yeah, the timeline is a little bit tight, so realistically, we got to it on day 5, no big deal. So I'm going to go through a note together with you about animals. There is a summary chart that you're going to complete in your notes on page 18 on vertebrates. And I just chose vertebrates because we're vertebrates and it's good to know more about vertebrates specifically. But you don't need to know all the phylums of animals. There are a bunch of them. Having sort of a general idea of what an animal is far more important than knowing specifically what the phylums are for this course. If you want to take a course in zoology in university, do it. It is absolutely fascinating. But this course isn't going to get super in-depth there. So there are some analysis questions that go along with it from the text 1, 2, 10, and 12 on page 107. So that's the animal portion. That won't take us long. Then there is a practice quiz. So keep in mind that the practice quizzes or the exit quizzes, I variably call them both things. They are the same though. Remember that those are simply to test yourself. The marks do not count. People are always asking me, do the marks count? Does it matter if I did poorly on the practice quiz? I mean, it does matter, but not for the way that you think. So it matters because it's giving you an indicator of some things that you need to go and review in your notes, or some things that you should chat with me about before you go write the unit test. If you don't understand something in the practice quiz, when you have a similar question on a similar topic on the unit quiz, which does count, you probably are not going to be able to answer that question. That would be my guess. So in order to get prepared and get to the level of depth that you need in order to be successful in this course, you are going to have to circle back if you find something that you don't understand in a practice quiz. That's the purpose of the quizzes. It's an alert mechanism for you. Hey, you probably don't understand this. Go back and take a second look at this or talk to me. Okay, so please do the practice quizzes. There are 2 of them. There's one after day 2. I think it's on the day 2 page. And then there's one today, yesterday, today, whatever. When we're done the main body of the content, we're going to do it today. So please do them. Again, they don't count for marks. I don't even check to see if you do them. That it is up to you if you want to check your knowledge. But just be aware that if you don't check your knowledge, there's a perfectly good chance that you don't have the depth of knowledge that you need in order to be successful on the unit quiz. And you won't know, probably. So again, I put these here for a reason. It's a pre-quiz, okay? It's to check to see if you have been following along and are comfortable with the concepts that we've been discussing. Okay. Okay, so that's sort of the content that we're going to be completing. I mentioned the summary sheet. Here it is down at the bottom. This is also on day 4 tab, but this is the summary sheet right here. You're going to download that and fill that in once we're done the animals section. Okay, and it's just asking you, like, for each kingdom, for example, for archaea, what type of cells are archaea? Are they multicellular or unicellular? And keep in mind that the answer could be both. It isn't for archaea, but it could be. So some things have multicellular and single cellular, like protists, for example. Does it have mitochondria or chloroplasts, or neither? How does it get its nutrients? And again, are they autotrophs or heterotrophs? Can they be both? You know, some things are both. How does it reproduce? Sexually or asexually, or both? What kinds of environments do you find these organisms in? And the other section is just, what are some other interesting or important things that we discussed about those particular groupings that you should probably note as you get prepared for the quiz? And some representative examples. Keep in mind that these should be species examples. Okay, again, I'm not going to check this, but this is a reference for you. it will certainly make the quiz a whole lot easier if you have this prepared in advance. And not to mention, it will force you to go over and review your notes, which will inevitably increase your understanding before you write the quiz, the summative quiz. Okay, so there's that. Like I said, I do have a completed copy available for you. It is posted right underneath. It says there's a completed version. Okay, I think I've got it going again. Can you guys let me know in the chat if things are running again and you're able to hear me and see me and I'm not super choppy? What's our status? Things look fine on my end. What about now? It's the same. Oh, but the bit rate went all the way back up again. Screen is blurry. Huh. So on my end now, it says that everything is fine now again. It says that it's receiving enough information for a normal stream, and my upload speed has gone back to normal again. Is it possible that you guys maybe on your end need to reload? Try reloading the stream on your end, like just hitting the refresh button. Those of you that are having problems. On my end, it's gone back to normal. Okay. Okay, cool. Sorry about that. Sorry about that. Technical problems. I still haven't run that network cable over. Oh, no problem at all. I want it to be clear for you. I'm still having occasional network problems, and I haven't run that Ethernet cable from my router or from my switch in the basement over here. So I'll probably do that. Maybe I'll do that during the break. I'll try and quickly make a quick Cat6 cable and run it over here, and then that should solve all of these connection problems. I think they're all Wi-Fi issues. So sorry about that. Okay, so I'm going to keep going. The things that you need to be considering today. It's the end of the unit. We're gonna finish the animals content in the summary sheet. During learning block 3, that's your time to work on the quiz. So you don't have to write the quiz during learning block 3. It's recommended so that in case you run into trouble or have questions while you're writing, that you can just pop into the Google Meet and chat with me, which you're welcome to do while you're writing. And my availability after school is mixed because I normally switch childcare responsibilities with my wife, who's been looking after both kids all day, which is a little bit of a crazy madhouse. I totally feel her pain on that. So anyway, I'm still available, but via email, and it's just not as responsive. So I recommend that you write the quiz during Learning Block 3, but you do not have to write it today during Learning Block 3. If you want, you can write it tomorrow during Learning Block 3, or you can write it tonight at 10 p.m. or like whenever you want, really. It's independent. It's open book. And so you're welcome to write it whenever it's convenient for you. There's just less support if you don't write it during Learning Block 3. And I want to support you while you're writing it. If you have any questions, you want me to clarify something, I mean, ideally, it would happen during Learning Block 3. But again, it's up to you. So do it whenever you feel it's appropriate. The quiz is open now, so you can really write it at any time. The deadline for it is tomorrow at midnight. And again, the deadlines are there to try and help keep you on track. If you don't follow the deadlines, you just end up stacking things, which is going to end up being a problem for you later. So I really strongly recommend that you don't do that you don't stack things, and that you make an attempt to hit the deadlines. If you're not going to hit the deadlines, just drop me an email. Again, things come up and I get that, which is totally fine, but just be communicative with me. And I would appreciate it if you were going to email me about not being able to make a deadline, if you just let me know what your alternative deadline is, because that's the first thing that I'm going to email you, which is, what do you think is a reasonable alternative deadline for you? And if you propose something reasonable, I'll have it in a day, I'll have it in 2 days, I have this soccer tournament, whatever. You don't even really need to tell me. It's none of my business what your personal issues are. I mean, you can tell me if you want, but I understand. Stuff comes up for all of us, myself included. But propose an alternative deadline, and then let's negotiate on that. And propose a real one. Pick an actual deadline that you can hit, and then I'm going to hold you to it. So what I've found is that when we propose alternative deadlines, students will often just say, yeah, probably get it done by whatever. It's like, no, you've misunderstood the purpose of this. I'm granting you an extension, which is totally fine, but it's based on a competent estimate in terms of your time of completion. Okay, so you need to hold yourself accountable to your alternate deadline. So pick a real one that you can actually hit, and then stick to that deadline. Otherwise, we're going to start running into trouble in this course, guys. Again, these are the constant problems with online only or even hybrid. I don't run into students every day. I don't talk to them every day. They start to get behind, and those people don't get the credit at the end, which I want everybody to be successful in here. So don't do it to yourself. Hold yourself accountable to your alternate deadlines if you have them. Okay? And if not, hold yourself accountable for the actual deadlines. That's preferable. All right. Enough about deadlines. The quiz is today. Write it today or tomorrow, whenever you like. I recommend Learning Block 3. The other things that are coming due are the 2 major assignments. The vocabulary list you've probably been completing as we've been going along, which is great. I mean, that's ideal. And then that's also due tomorrow at midnight. But ideally, you would just hand it in today whenever you're done today's content. You're done the animals part, you're done. So fill in the rest of your things in your animals vocabulary list and submit the assignment. So ideally, again, you've been completing it all along. If you've left it to the end, that's on you. You shouldn't have done that. I really strongly suggested that you didn't do that, but you are going to have extra work now to do at the end of the unit. It's not meant to be how the assignment's completed, but sorry, you made it hard on yourself. Don't do it again. The last part here is the species spotlight assignment. As I mentioned, I'm going to give you a bunch of time to work on it today. And it is also due tomorrow at midnight. These things always come due at the end of each unit. We've gone over what that entails already, so work on that at your leisure. Okay, so let me talk about the animal content real quick here, and then I'm going to stop talking and let you guys get to work. And then in the second block, you will have a little bit of time to work on your species spotlight as well. You will this block as well. And then sort of towards the end of that, we're going to get into the next unit. We got to start already, which is genetics. Okay, so animals. How do you know you're looking at an animal? Well, they're eukaryotes. I mentioned this idea yesterday, but there are some protists that are colonial. That is, they are not multicellular, but they are single cells that can operate in a multicellular way. Those are called colonial protists. I mentioned the slime molds yesterday. And those are thought to be the ancestors of animals. Basically the first multicellular motile organisms, okay, that can move around. If you look at them and compare them genetically to the oldest phylum of animals, which is the Porifera or sea sponges, they have some genetic characteristic similarities. There's also quite a bit of difference, but you can see how there's a relatively close common ancestor between the two of them. And so that's our closest connection. There's probably a bunch of extinct species sort of in the middle there. But again, we don't have genetics for a lot of those species and stuff. So we're kind of playing a little bit of a guessing game. We have the genetic data of what's alive now and recently alive, and then we kind of have to fill in the blanks a little bit in the middle. So that's the thought anyway, is that we're related to colonial protists. That's probably the closest grouping. All animals are multicellular, so there's no single cell animals. If it looks like an animal but it's only one cell, it's a protist for sure. They're all heterotrophs, so you're not going to find an animal that is autotrophic, makes its own food. Most animals are aquatic, which may be surprising to you. We're not aquatic, and so we don't tend to think about diversity as being much more common in aquatic environments than terrestrial, because we live in a terrestrial environment, and so we tend to only think of the living things that we see around us, which makes perfect sense. But in terms of diversity, the vast majority of animals live in water. So we also evolved from water, so animals were obviously present in water long before they were present on land. So anyway, that makes sense. That's where our ancestral origin is, and it's also where our closest related grouping outside of animals, which again are colonial protists, they also live in water. So seawater, salt water, is again the most common place to find an animal of all kinds. Other than that, there are slightly fewer in freshwater, but freshwater ecosystems are the next most diverse ecosystem for animals. And then land is the least diverse, although again we tend to think of it as being diverse because we're from the land, but it is, in terms of pure numbers, by far the least diverse place to find animals. Animals are characterized by being motile, so every animal is motile. They have the ability to move themselves or objects around in their vicinity at will, at some stage of their life cycle. So I didn't write that on there, but that it's key to say that they are mobile. Oh, I did write that. I wrote it right below here. They can move at some point in their life cycle. So all of these groupings, all of these phylums are motile for their entire life cycle, except for Porifera, sea sponges, which are only motile during their infant stage, basically, where they're floating around. I shouldn't say floating, they're swimming. It's directed movement. But then once they attach to a place on the ocean floor, then they've essentially become sessile for the rest of their life. So, but they are motile for part of their life cycle. The same thing is true for some cnidarians. So I'm kind of overgeneralizing here. Some cnidarians actually attach themselves to the seafloor, in which case they become sessile for a large part of their life cycle as well. But they all have a part of their life cycle where they're motile. Okay, so those are the general characteristics. This is a table right here of animal phylogeny. So it's showing all the phylums of animals. Oops, didn't mean to delete that one. So all of these things are animals, and probably the one that sticks out the most for you is the most ancient of all animals, the Porifera, because you probably didn't even know it was an animal. I'm not going to lie. In high school, before I took this course, I had no idea that sea sponges were animals. They look like plants, very much. They do move. They're motile in some parts of their life cycle, but they also have the ability, some of them sway. They're filter feeders, so they pump water through their inside, so they're able to move their natural environment around as well at will. But they are alive. So, I mean, no, I guess it's obvious that they're alive, but they are animals. Now, I should mention that we are, and some people were asking about this yesterday in the Google Meet, we arbitrarily decide what is and is not an animal, right? Like, we picked those characteristics, but based on the characteristics that I just mentioned here above, sea sponges fit those. So you could say, you could call it a colonial protist. We could have cut them off and said, well, you know, they have to be motile for their entire life cycle. So then we just cut it off here, and then that becomes a colonial protist. We add it to the phylogenetic tree for protists, stemming off of the colonial protists. And again, genetically, that's a pretty close relation as well. So you could categorize this slightly differently and change the system up. But again, we picked these subdivisions here. And based on the subdivisions we chose for animals, they are animals. So how do we generally organize animals? These are the phylums. We split them based on sort of 4 major categories that we're using to subdivide these. Okay, and I mentioned these way back when we just were talking about dichotomous keys. I guess this is a sort of a form of dichotomous key. It's not really a very good dichotomous key. It's not. These are not all yes/no decisions. There's other things here that have no decision points, so it's not technically a dichotomous key, but it's a similar idea, which is that we look at body layers first. So that was one of the first things to evolve in animals, multiple body layers. Porifera, sea sponges, do not have multiple layers. They have one single tissue that runs through the entire plant. So if you looked at yourself, you have layers to you. You've got skin tissue. You've got subdermal fascia, nerve, muscle. These are tissues, right, that are different as you go through the organism. Porifera do not have that. So they are one continuous tissue from the outside to the inside. You don't like find different things in them. It's consistent all the way through. They don't have epithelial tissue on the outside with some kind of connective tissue on the inside. It's all one tissue. Okay, so that's one thing, and we can also split groups up later on based on the number of tissues that they have, and some of these groupings are based on the number of tissues. Animals typically have the 4 that you learn in grade 10, which are nerve, muscle, connective, and the other one, epithelial. Okay, so we did mention those as the 4 animal tissues in grade 10. Most animals have those, but not Porifera. We can also separate them out by whether or not they specifically have nerve tissue. So that is really the first split. Porifera have no nerves. All the other groupings of animals have some form of nerves. Even cnidarians, which are jellyfish, have a nervous system. They don't have a central nervous system, which means that they have a one spot on their body that is sort of like the coordinator of all nerves, which you could sort of think of as a brain. It's not, in other animals, it's not developed enough to be called a brain. Sometimes it's called cephalization. It just means that there's like an area of nerves, like a pocket where there's more nerves. That's cephalization. But it's not really like a true brain, because like not everything is coordinated at that spot. But cnidarians don't even have that. So they have a distributed nervous system where the nerves are just kind of around and they communicate with each other, but there's no like central spot that's like, this is where the decision maker is. So cnidarians don't have that. They do have nerves, but it's not centralized. So we also split them up based on the idea of symmetry, and I mentioned this as well in the dichotomous keys lesson, this idea that peripera, sea sponges, and cnidarians are radially symmetrical, and pretty much everything else in the animal kingdom is bilaterally symmetrical. So bilateral symmetry is a very early evolved characteristic. Okay, all these ones on the right side here are bilaterally symmetrical. You can also look at their embryonic development. Okay, so like how does their digestive system build during embryonic development? And again, that only applies to these over here. It doesn't apply to these two because these two don't have a digestive system, not a traditional digestive system that's like a tube that goes from end to end where you like process food. Their digestive system works quite differently. So you can't even classify these based on that, which is why that that classification you don't even find on the other side. So, but anyway, you can look at whether or not the digestive system develops in the embryo from mouth to anus or anus to mouth. The oldest ones develop from mouth first. Those are called the protostomes. So those are the older animals, and they're on the left-hand side here. And then the more recently evolved animals, the deuterostomes, develop the other way from anus to mouth. Okay, and that sounds like something relatively minor, but oftentimes small differences at the embryonic level during development result in large differences at the developed level, which in this case is true. So that affects a whole bunch of stuff about coordination of the digestive system and stuff, which is important as a developed mature organism. Okay, so we categorize them based on that, and then you can also look at the germ layers during embryonic development. When you look at a developing embryo, you can see that there are layers of embryonic tissue. And the number of layers that you have determines the type of layout that your body will have at the end. And so we typically have 3 germ layers. One forms the outer structure, skin, bone. One forms the main bodies of the organs, and then the other one forms the digestive tract in the center. I'm hugely overgeneralizing here, but there's 3 layers. Okay, but some of them are simpler than that and have fewer, and so you can use that to subdivide these as well. Okay, obviously more layers is a more recent evolutionary development. So that's just some basic stuff. That's how we sort of differentiate between them from an evolutionary perspective. And you can look at the groupings over here. What we used to do in this course was crazy, which was that we would spend, I don't know, a long time, a day, and we would just look at 4 of these groups and talk about them in detail, and then spend another day and talk about these 4, and then spend the last day and just talk about the last 2. We spent a long time on chordates because we're chordates. So we'd really, really dig into the animal phylums here, but that we just do not have time for that anymore. It's a cool idea, but that we don't have time for. So anyway, there's nothing for you to fill in down here. I actually removed this question and I forgot to get rid of these lines, again, due to time. But having a general sense of the types of animals that are available, that exist in nature, is probably a good idea. Peripher, sea sponges, cnidarian are like, I mean, they're not all jellyfish, but they're jellyfish-like. Platyhelminths are flatworms. Rotifera are, how would I describe a rotifer? They're ocean-bound. I want to call them tube worms. That might be actually what they're called. They might be called tube worms. Mollusks are a group unto themselves. They're mostly bivalves. They are filter feeders. Annelids are called roundworms. Earthworms are an example of that. Nematodes are another type of worm, a totally separate group to annelids. They are often parasitic. Arthropods you're probably familiar with. There's a subgroup of arthropods called insects, which is something you're definitely familiar with. I'm sure you've heard of an insect before. Echidnoderms are things like starfish, although there are various types of echidnoderms. And chordates are the most recently evolved grouping here, things that have a spinal cord. Okay, those are the basic groups. Again, you don't really need to know them. I'm not going to quiz you and say, like, is this a phylum of animals, or which 3 are phylums of animals? I'm not going to ask you that. Although keep in mind that you have access to your notes during the quiz. So, we're going to look just at the chordates, okay? Again, normally we would look at each of these, but I'm gonna zoom in just on the chordates for a second, and we're gonna talk about them in more detail. And the reason I chose that group is because we are chordates, and it probably pays for you to know a little bit more about things that are like us. Okay, although it's probably important to know about all of them, but we don't have time. So, most chordates are vertebrates. So a vertebrate is an animal that has a spinal cord, a cord, making it a chordate. Okay, chordates just have a spinal cord. Most things with a spinal cord also have a bone protecting the cord. Okay, so if you have a bone protecting the cord, we call that a vertebrate. There are some chordates that don't have a bone protecting their cord. So this is a common mistake in that people assume that chordate and vertebrate are the same thing. Most chordates are vertebrates, but not all. So there are a couple groupings, a couple classes of chordates that are not vertebrates. We don't really care what they are for this course, so I'm going to ignore them. They're all ocean going animals that are not super relevant to this course, but most chordates are vertebrates. Okay? When we say that something's a chordate, again, I mentioned that it has a spinal cord. It means that during embryonic development, you can actually see a cord of nerve tissue that goes sort of from the top of the organism to the bottom of the organism, and that's called a notochord. So a notochord is like a primordial pre-version of a spinal cord, and it becomes a spinal cord. Well, it becomes a cord. I'm not sure if I can call it a spinal cord, because spinal implies that it's inside of a vertebral column. So anyway, it has a cord. There's a nerve cord in there called a notochord, okay? So the notochord is the cord, but vertebrate implies that there's a bone around it. Invertebrates are things that don't have a backbone. So there are some chordates that are invertebrates. Not many. There's only, I believe there's 2 classes of chordates that are not vertebrates. They're relatively unimportant. But all of the other animals here, so all of these other groups right here, except for most chordates, I overlap a tiny bit there because there are some chordates that are invertebrates, but all the other ones you would call invertebrates or invertebrate animals. Okay, so when we're talking about invertebrates, it's a very broad group. It includes everything in the animal kingdom except for a select group of chordates. Okay, so that's what we mean by invertebrate. Most animals are invertebrates, obviously. Okay, so invertebrate is by far the most common. And again, you can, we just talked about the various groups here, but the simplest ones are sponges. They have only one tissue. But peripera are also fairly simple. Peripera are sea sponges, but hydra and jellyfish, which all are cnidarians. Hydra and jellyfish are both cnidarians. They're also relatively simple. So they would be on the real simple end, the far end one way, and then the far end the other way is where you would find the most complex organisms. They're pretty much organized that way here. What I'm going to get you to do is read 104 and 105 in the text, which is a very basic overview of vertebrates. Okay, again, we're going to focus on the group that we're a part of here. And I'm going to get you to fill in this chart. So these are the different classes of vertebrates. Okay, they include agnathans, which are jawless fish, Chondrichthyes, which are cartilaginous fish. Okay, those are the ones that you cartilaginous fish. What would that be? Like lampreys? Oh boy. Actinopterygii. Actinopterygii. I think Pterygi? Actinopterygii. Tergi. 2 eyes is eye. Actinopterygii. Well, I'm probably butchering that. Those are bony fish. Sorry, a lot of these groupings are things that I am not running into on a regular basis. Actinopterygii, I think is what it is. Or bony fish, which, again, you would be familiar with. That'd be like a trout or something like that. Amphibians, which you're probably familiar with. Reptiles, which you're probably familiar with. Reptilia. Aves, which are birds. And mammals or mammalia, which we are a part of that class. So I'm going to get you to go through, provide 2 examples of each. You may have to look these up. Oh, lamprey are acnathans, so they're not chondrichthyes. I'm not sure. Which ones are chondrichthyes? And sharks? Oh, sharks are probably chondrichthyes. Yeah. Because they're cartilaginous fish, right? They don't have bones? It's all cartilage? No, I'm going to double check that. I'm not 100% sure. Anyway, 2 examples of each. I should probably go fill this table in myself. 2 key features of each group. And the number of known species. That you can take directly out of the textbook. You don't need an updated number from online. The one in the text is close. It's probably a little on the low side because we've definitely discovered more of these since then. But some have also gone extinct, so there's a give and take there. Stingrays are cartilaginous too. Okay, so they would probably fit under chondrichthyes then. Okay, noted. Thanks, Andrea. And there are some questions that go along with it from the text. Okay, it's mostly based on actually what we were just discussing right here. So that's the basics, what you need to know about animals. We're not getting super deep into this. We're not going to spend a class looking at each group, which it would be so cool. I mean, we could make a whole course out of zoology, but no time. So like I said, I'm going to get you to fill out this table. So what we're doing right now, you are completing that summary table and the analysis questions. There they are. They're shown on the day 4 tab because this is sort of what we're picking up from yesterday. You want to add any new words to your vocabulary. This is the last lesson for this unit. So once you've gone through and added your vocabulary, you're done the vocabulary list, so you'd hand it in. Try the practice quiz. And then the last component of this is try and fill in that summary sheet. That summary sheet is meant again as a review for the quiz. So you could do it now. You could do it right before the quiz if you feel that it would be helpful to do it before the quiz. I'll leave it up to you when you want to complete the summary sheet, but you should complete it. It will help you do a review of the different groupings so that it'll have it more fresh in your mind for when you're writing the quiz. Okay? So that really completes the unit for us. The last aspect of this is that you need to work on your species spotlight assignment. So if you finish this stuff up early, you want to move on to your species spotlight assignment, that's great. I mean, you probably should have some time because that stuff won't take you that long. I shouldn't say that. The summary sheet might take a minute, especially if you don't remember some of the groupings that clearly and you need to go back and review your notes. It might take this time. That's fine. I will give you, hold on, let me double check to see how long the first lesson of genetics is. Cell organelles. Oh yeah, it's pretty short. So, okay, good. That was smart then. I kept it relatively short. So, I'll give you half of the block 2 as well today to finish this stuff up and work on your species spotlight assignment. And then the end of block 2, maybe with about an hour left, I'm going to get into the beginnings of genetics. And the beginning of the genetics is actually just a review of grade 10. We're going to talk about the cell organelles, which hopefully you have some memory of those, mitochondria, ribosomes, etc. Just to jog your memory on what they all do, but you do need to know that for genetics. And then we're going to get a little bit into just the beginnings of what is genetics, and what is DNA and stuff like that. So we're just going to start that towards the end of today. Okay, so I'm going to stop talking. I'm going to let you get to work on this stuff. Keep in mind then again what is coming due. species spotlight assignment, vocabulary list, and quiz. There's always 3 things, always, and they're always due the day after the end of the unit at midnight. So all of them come due tomorrow at midnight. That's the day after the unit ends. If you're looking for the assignments, they're on the assignments tab. If you're looking for the quiz, they're on the quizzes tab. Okay, but you'll always be able to find them. If you have an accommodation for extra time when you're writing the quiz, I have already gone through, I've reviewed everybody's IEPs, and I've already added in that extra time. So it should say for you 2 times the time, or you'll be able to see it there when you access it. If you have any problems with that at all, something's not working, or you believe that you should have an extra time accommodation and it's not there, please contact me. Okay, I believe at this point that I have set it up correctly. So, so I've made an error then if it's not, and you need to let me know that so I can fix it for you. Okay. Any questions about what we've just covered here? Well, really anything. The quizzes, the assignments coming due, anything? I'll wait for a sec here. Give people a chance to type if you need to. Whoop. Oh, excuse me. Allergies. Oh, 11:05. I will see you guys again at 11:05. Normally, by the way, when I leave the live stream, if we are going to come back, and we generally are, I will put that right on the little screen. But it's 11:05, so I'm going to let you guys work right till 11:05. Don't forget to take a break for yourself. And we'll come back, take attendance. I'll probably send you guys away again for a bit to complete what you're working on, because there's a fair amount of stuff there. And then I'll also let you know. I'll post it up there, like we're coming back at this time to get back into genetics. But I won't surprise you or anything. Okay, if that is the only question, then I'm gonna let you guys get to work, and please let me know if you need anything. I'm available the entire time here. If you wouldn't mind, please letting me know in the comments that you are present for the second half. One thing I wanted to mention really quickly. Oh yeah, that's right. The other day I mentioned the idea of going out in the back and getting some protists and showing you some examples here with the microscope. So I did do a little Amazon perusing to look for some supplies. And sad to say, there is not a really great source to overnight some microscope equipment to your house. I don't know why I thought that would be a thing. You can buy stuff on Amazon, but it is insanely overpriced. $50 for 100 glass slides. That would be 20 times the market rate. Glass slides are very cheap if you buy them from a proper supplier, which takes quite a while. But Amazon is not the place to get that stuff. But it is the only place to get it quickly. So I think what I'm going to do instead is I'm going to contact my boss and then find out if it's possible for me to just swing by the school quickly and pick up some supplies. In which case I can grab some microscope slides, some dyes, some methyl cellulose, so that I would be able to show you some of this stuff at home. I already have a microscope. I own a microscope, which is, I guess, kind of nerdy. It's not the best microscope in the world, but it works fine. But I don't have slides and things like that. I have some prepared slides, but nothing that's useful for this course. So anyway, I'm gonna get on that. investigate. Maybe I can swing by tomorrow after school or something. I got to plan this, but I'm gonna try and get that in during the first block, so we're able to look at stuff. I'll also see if I can find some of the prepared slides at school that we have for plants and a few of the other things like from the other units, so that I can show you some live views of some of the tissues and things like that. So I'll investigate that, but I won't be able to overnight it the way I thought I would. I thought, oh, I'll just spend a couple bucks. I'll spend $5 on microscope slides, and no, there's no such thing as that. Apparently, Amazon has a crazy markup on that. So, not a great place to buy microscope slides. Okay, cool. Everybody's here. So, as I mentioned, I'm gonna give you some time to keep working here on you guys are, I'm assuming, done the animals component at this point, but filling in your summary chart, which is really preparation for the quiz. Why don't we say, and working on your species spotlight assignment. So and that's going to take a significant chunk of time. So why don't we say we'll come back at 12:15, and that should give us enough time to cover everything that we're going to start in the genetics package. So the first day of genetics, like I said, it is a lot of review, mostly about cell organelles. And I realize that people's experience in grade 10 has been somewhat variable, in that some people kind of pieced out at the end of last year. So they may have taken grade 10 science and then maybe didn't really do the biology unit, or depending on where you were in your course. Some people did, some people didn't. So some of this is possibly new for you, I guess. It is part of grade 10. So we'll obviously, I'll be there to support you if you're like, I have no idea what's going on with these organelles, and I can give you extra resources if you're in that boat. But we'll do a review. Anyway, basically what we're going to do is go through a review of organelles, the idea of cell theory, which, again, should be reviewed from grade 10, but may or may not, depending on the circumstances. You guys are going to label a cell. And then I've got 2 quick little videos here that are an introduction on how DNA is used to make stuff. Because DNA is a molecule, which I guess probably is not surprising to anybody here. But how do you go from having a molecule to making something? I mean, that's a pretty big step there. Like, how do you use a molecule as an instruction manual to build something? So we're just going to scratch the surface of that process today. We're going to talk about how it works in general. You're going to watch a couple quick videos, and then tomorrow we're going to get into that process in a lot of detail. So we're really going to dig in. We're going to talk about the biochemistry of a little bit. We don't get into the biochemistry too deeply, because this is an entire unit in the grade 12 course just on DNA transcription and translation, or using DNA, where you get into really all the fine points, and you need to learn all the different enzymes that are involved and how each enzyme works. And so we're not going to get into that depth, but a pretty good overview of the process and how it works. And the reason that we're doing that is because that is the basis of genetics, which is that you take a piece of DNA and then you make something out of it. So you have to have a basic understanding of that process, how you go from DNA to a thing, in order to sort of understand all the other stuff that we're going to talk about in genetics, which is all related to that process. So, like I said, we're going to get to that today. I'm going to come back at 12:15, and then we'll dig into it. But not a tremendous amount of detail today. Then the next day, tomorrow, we'll get into this process in a good deal more detail. And then we're also going to talk about how you make gametes. That's tomorrow as well. So I'll let you get back to work here. If you have any questions from anything that you've been completing so far, the animal stuff or whatever, don't be afraid to put it in the chat. If it's something that you want to have an interactive conversation about, please come to the Google Meet at 1 o'clock. And again, I have my whiteboard available here. I'm able to discuss stuff with you, give you more details. And you know what? People have been coming to the Google Meet just to chat about something that they're interested in biology. Actually, several people have done that. Cool. I love biology. So stop by anytime. We can always talk about any particular thing that piques your interest in biology. I don't necessarily always have the answers. Sometimes I've got to do a little bit of research on my own. But I'm happy to talk about anything that you're interested in. If you want to follow up on a particular topic or would like more information about a particular topic, I can find reading material for you. There's often cool YouTube videos. If you have any interest in any of these topics in biology, again, a big part of this course is to sort of help you find something in biology that is important to you, and maybe get you into biology a little bit more. And I'm happy to do that. That actually makes me very excited that students are interested. For the assignment, do we have to choose a species that we talked about in class or choose anything? You can choose absolutely anything that you want, as long as it does not have a vertebrae. Okay, no bone around the spinal cord, you're good to go. I recommend that you choose non-vertebrate animals or protists or fungus or plants, and that you probably stay away from the prokaryotes, bacteria and archaea. And I only say that because it's harder to create a dichotomous key for bacteria and archaea. It's not impossible. We have them. So you can do it. It's just more difficult because they pretty much all look the same, especially the closely related species that you would put next to them on a dichotomous key. They're going to look identical. So, and you can do that. You can talk about their biochemical properties, certain genes that one would have and another one wouldn't have, or whatever. But it is more difficult. So again, you can go that route. Other people have. Actually, I take that back. I'm not sure if anybody's done a prokaryote before. Not this year. In previous years, I've definitely seen them, but I don't think I've seen one this year. But anyway, it's up to you. But anything that's not a vertebrate, maybe I've never mentioned it. There's a ton of diversity out there, so you pick. Pick something that piques your interest. Okay, I'm going to let you go now. Stop talking. And again, see you at 11:15. I'll put it up on the screen. Okay, I will get started now. As promised, it's 12:15. So what we're going to discuss now is, well, an introduction to the genetics unit. Keep in mind that this unit is just like the first unit in that there is a vocabulary list. Okay, so you start a fresh vocabulary list for this unit. There is going to be a summative assignment that is due at the very end of the unit, which we'll talk about a little bit later in the unit. It doesn't really make much sense to talk about it now because we haven't covered anything that you're going to discuss in the assignment, so I'm not sure it'll make much sense at this point. But basically, you're going to create a learning tool to teach a concept to a fellow student, and you're going to do it in the form of a video. People always get all nervous about making a video. You don't need to show your face or anything. You are going to narrate it, so you are going to explain a concept, but you don't have to show your face or anything. So, and we'll talk about some technologies you can use. I already mentioned the idea of Loom or whatever, but some people use Vidyard or whatever. That's also possible to use as technologies that you can use for your learning tool. I would call it a video or a presentation, but it's I've had people do all kinds of different things, and it's pretty open in terms of like how you want to format it. Some people have done stop motion animation. There's been a bunch of things. So anyway, just be aware that that's coming towards the end. We will discuss it in more detail after we've covered the concepts that you're going to be discussing in the presentation. So, but the vocabulary list you can get started on right away. Keep in mind that going out of focus. Keep in mind that you need to make a new homework document as well for this unit. So it's a separate Google Doc for your textbook homework for each unit. So when we get to the textbook homework for this part, you will need to create a new homework document. I don't think there's any textbook homework from today. There's stuff in your notes, in your course notes, but nothing in the textbook. So you probably wanted to create that document today. So I'm going to start off discussing a very brief overview of cell theory, which you talked about in grade 10. And I'm going to give you a little bit of work to do in terms of describing the various organelles of the cell, which, again, should be a review for you, but it may not be. You're going to need this notes package, though. So if you're looking for the notes package, there it is. I believe it's also on the sidebar. Yeah, there it is. Unit 2, Part 1, Review and Genetic Processes Course Notes. The course notes for this unit come in a couple smaller packages, so I'll let you make a copy of that and open it in Cami, or if you're not using Cami, if you're using some other type of editing software, sure, no problem. Or maybe you printed out the notes. Again, it'll just be the next thing then in the course notes if you printed them out. I'll give people a second to open that up, and then let's get right into it. Oh. Is this gonna... Oh, there she goes. Too slow. Let's restart it. This program used to be relatively lightweight, but now it's become a real memory hog. I think they've kept adding features to it. Here it is. Okay, let's talk about the cell. If you can imagine, only a few hundred years ago, not very long ago, we didn't know anything. I mean, really, we didn't know anything about living things. I'm not even going to try and mince words on that. We knew very, very few things about why things were alive and how living things functioned. There were some anatomy books that looked at the macro structure of animals, including humans. Some of them were made by Leonardo da Vinci, actually, that were quite detailed. But even in that case, a lot of misconceptions were transmitted from generation to generation. So let's just say that those texts are problematic, and certainly our understanding of how those systems worked was fairly poor, up to even 300 years ago. So we were talking about like post-Renaissance here. And part of the problem is that on the smallest level, we had no idea what anything was made out of. Really. I mean, look at yourself without the aid of a microscope and just like look close. It doesn't really tell you very much. You can't really see what's going on there. Why is your skin alive? And in order to actually figure that stuff out, you have to be able to look at stuff pretty up close. So early 1700s, we're starting to develop the first real microscope technology. And as a product of that, we developed what's called cell theory. Cell theory is our idea, our current understanding of how things work on the micro scale, okay, below the level of what the human eye can see. So sometimes these first three rules are known as the classical interpretation of the cell theory, because these are like sort of like the first things that we discovered about cells, okay, through early experimentation and observation. So the first 3 points here are the first 3 tenets of cell theory. All things are made out of cells. And that goes back to our first unit where, everything's made out of cells, right? All the living things that we just looked at in all the different kingdoms, if you look right down to the smallest level, they're made out of cells, or a cell, single cell for several of them. Viruses are in some kind of weird gray area, but all the living things that have all the features of living things are made out of cells. Okay, and living things can be uni or multicellular. That's the first tenet of cell theory. Everything that we've ever seen that exhibits the characteristics of a living thing is made out of cells. Those cells are the structural and functional units of life. That is, if your body does that as a multi-celled organism, all it is a product of what your cells are doing. It's kind of weird to think of it that way, but like you're having thoughts right now. It's a weird thing to connect that to what your cells are physically doing, but really it's just a product of all of the neurons functioning as cells in your brain. And how do you go from having a number of neurons functioning, nerve cells, to consciousness, being awake and alive and self-aware? And we don't actually understand that. That's on the barrier of our current understanding of neuroanatomy and neuroscience. We don't know the answer to that question. That it's a really cool, interesting question, though. So but it's still true, whether or not we understand it. Our consciousness is just a compilation of all of the different cell activities that are happening inside of our brain. We consume food and break down food, and that's just the product of all of the different cells of our digestive system doing their jobs to nutrients. And we'll get into that in more detail when we talk about animal systems. But anyway, the smallest structural and functional unit is the cell. If you go smaller than the cell, you start to lose some of the structure and function of life. So sure, mitochondria can convert certain substrates into ATP, an energy molecule, but they can't survive on their own. They can't acquire their own food. They can't dispose of their waste. They can't grow on their own. So if you go down one lower level to the organelle level, you can't separate them out and have them be structural and functional on their own. So that's what it means by saying that the cell is the structural and functional unit. It's the smallest structural and functional unit of a living thing. And as far as we know, cells always come from pre-existing cells. In other words, there's no such thing as spontaneous cellular generation. And I mean, I guess this just makes sense. You're not just going to have cells just appearing out of nowhere. Now, of course, there was a time in the early days of biology where we didn't know that. We didn't know where cells were coming from. We thought when you got microbes in growing in a sample of water or something like that, we assumed that they just appeared, put themselves together from the surroundings, which is a fascinating idea, but we've never observed that before. Now, obviously, that's problematic, going back to what we were talking about in the diversity unit. If you go back through the phylogenetic tree and you trace the tree back and back and back and back and back, and all of a sudden you get to the origin, the first cell, and then, line. Where did that cell come from? If all cells arise from pre-existing cells? Great question. So I think I mentioned this back in the diversity unit. We don't really know the answer to that question. It's possible that the original cell structures may have originated not on Earth. I mean, sure, could have arrived on a piece of debris from some other place. Who knows? We don't know where the original cell came from. We have some great experiments. I mentioned this in diversity, I'm sure I did, that demonstrate that you can build a lot of the building blocks of living things just out of the materials that were available in the early Earth, and they do spontaneously generate. So this last point here is sort of like yes, we've never observed this, but I mean, the implication is that it did happen at one point. But we've never been able to replicate it, and we've never observed cells arising from no cells. So that one's a little bit shady of a point. We've also added some things into modern cell theory because we've been able to look at cells a lot more closely and at their biochemistry. And that is that there is some type of energy flow occurring within cells. Cells must take up nutrient of some kind from their external environment. They must do some chemistry on those nutrients and derive energy from them. So there is an energy flow that exists within the cell, and then their waste products are exuded. So that must be happening and is observed within all living things. The hereditary information, so how a cell passes on its data on how to build itself and how to do all of its functions to its daughter cells, is contained within a molecule called DNA, which we're going to get into more detail about in this. Are we going to get to that today? Yeah, we might just start chatting about it today, but we'll get into far more detail tomorrow. And it's passed from cell generation to generation. So you are going to pass, or I mean, you could, if you wish, pass on half of your genetic information to a child, if you wish, if you're capable. And all organisms do that. If you're asexually reproducing, you're passing on all of your DNA, potentially to an offspring. And you're making an exact copy of yourself. That's clonal reproduction. But it's also possible that you sexually reproduce, in which case you only get to give half. So that's the information that's getting passed on. We know now that it's DNA. And we only figured that out relatively recently, by the way, early 1900s. Before then, we had no idea what it was that was going from parent to child that was allowing them to, get characteristics from their parents. We thought maybe it's protein or maybe RNA, or there's a bunch of candidates that we had to figure out eventually that it was DNA. And the last point here is that cells have the same basic molecular composition. We're going to get into this in the digestion part of the animal systems unit as well, because we're going to look at where you get all of the different parts that you need to build cells from your food that you eat. But they have the same basic molecular composition. I mentioned DNA already. They all have RNA. They all use fats to build their cell membranes. They all have certain enzymes that are responsible for zipping and unzipping DNA, for reading and writing RNA. Ribosomes are shared. Anyway, the basic molecules that allow cells to operate are shared between all living things. So I realize that when you look at a bacteria under the microscope, it seems completely foreign to you. That is completely different an organism than us. You would almost think that there's nothing the same, except for the fact that their cellular machinery is, similar, certainly. And some of them are identical, but a lot of it is similar cellular machinery. So the basic molecular stuff is the same across all living things. Okay, so that's the idea of cell theory. What we're made out of at the smallest level, and that's sort of what we know. Okay, obviously we know it in a lot more detail than that, but that basic premise has been proven, or is currently accepted based on the evidence that's available, I should say. That's how theory works. That's a weird sound. What we're going to talk about here when we talk about genetics and cellular composition and stuff like that is primarily eukaryotic. So, yes, prokaryotes are made of cells, are single-celled, and they have organelles. However, the way that they do things is a little bit different than eukaryotes, just because they have different structures that are involved and stuff like that. And so we're really not going to touch on how prokaryotes do their genetics. If you're interested in that, you can take whole courses in that in university. You can do a whole degree in that if you're interested. But we're going to stick to eukaryotes for the most part. And the reason for that is we're going to try and apply some of this stuff to people. And the easiest way to do that is to focus on eukaryotes and like what this would look like in us. Okay? It is similar in prokaryotes, but there are a lot of significant differences that you can't transfer this information directly over. And so I'm just giving you a heads up straight from the start. We're going to focus on eukaryotes. We're going to talk about us, basically. We're also going to mostly focus on animals because there are minor differences here between plants and fungi and animals in terms of their cellular composition or their normal cellular composition. And so we're going to focus mostly on animals. I might mention some stuff that fits with other groups. But again, I'm going to keep this mostly to animals. Otherwise, there's so much stuff to discuss in terms of differences that it's going to make this far, far too complicated. So that's fine. We're just going to focus on animals. I am going to get you guys, when we're done here, to read through this. Okay, so this is just a summary of what an animal cell has inside of it and what each of those parts does. Okay, the organelles and their function. It's framed as a coloring book. Coloring books are a memory tool that works for a lot of students. So for example, you go through, you read about the nucleus, and then it says, color the nucleus brown. So you go down to your cell, you find the nucleus, and you color the nucleus brown. Okay, and while you're coloring it, the idea is you are thinking about and recalling all of the information that you just learned about the nucleus. Oh, there it is, and there are the nuclear pores, and this contains all the genetic information for the cell, and okay? You don't have to do this. This is a memory tool. I will ask you to, you still need to read it, and you still need to label your cell, but the coloring component is really, I'll leave that up to you. When I was in university, I bought a coloring book. We were asked to buy a coloring book for learning human anatomy. And it worked exactly the same way. So you learn about a group of muscles, and then you go through and you color them, and then you look at where the attachments are, and you think about it. And it's like, oh, this muscle is the blank, and it moves the middle phalange, or whatever. And the idea is then you go back, and you can color over it, and look at your coloring again to help recall and memorize all of the different anatomy in the human body. Okay, we mostly did muscle and bone in that course, but so it was musculoskeletal anatomy. This is a great tool for a lot of students. There are many of my friends that told me that was how they got through anatomy, was that coloring book. For me personally, I found it completely useless. I used it, and it did absolutely nothing for me. My strategy that works for me personally is I'm a flashcard guy. So I made flashcards. I looked at them. Then you look at the picture, and it's like, can you name them? No? Okay, go back and look at the other side of the flashcard. Move on to another thing. And then you come back around, and then the next time you try and name them, can't get it, look on the back of the flashcard, and then eventually you're going to come around and you will be able to name them. And then you just do that every day. And eventually you build up a big pile of flashcards, and you know all the anatomy. So that worked for me. Okay, that was my personal strategy. That worked for some other students as well. But the coloring book did nothing. But again, other students swear by it. So I'm not going to write it off as a memory tool that is non-functional, because it definitely works for some people. So I leave it here for you as an option. If you feel like the coloring while you're thinking about this stuff is going to help cement it in your memory, great, then definitely use it. You can buy these biology coloring books for pretty much any topic. You can get a cellular biology coloring book. You can get a enzyme coloring book, all kinds of stuff. You can even get ones for chemistry knowledge and stuff like that, where you color facts and things like that. There's all kinds of memory tool books out there. Again, it's not my bag, but if this works for you, then color them in as you go. Okay, but I'm not gonna force you to color them. I'll leave it up to you. So what you're going to do is read through this, color if you want, and you are going to need to label them as well. Okay, because you do need to know what the parts roughly look like. Hopefully you learned this in grade 10. And you do need to know their function. Because later on, as we go through this process of transcription and translation, I'm going to refer to these all the time. And the expectation is you have some idea of what I'm talking about when I refer to them. Okay, where they're located, what they look like, relatively. Okay, you don't need to have a in-depth knowledge of each, but you need to know the general function. The nucleus is where the DNA is located. It has pores that allow RNA to go in and out. The nucleolus is where RNA is produced. The rough ER is has ribosomes attached to it. And it is responsible for processing completed proteins or completed polypeptides and transporting them around the cell, sometimes to the Golgi apparatus. The Golgi apparatus is responsible for folding proteins into their final shapes and packaging them for exporting them out of the cell. The mitochondria is the primary energy production site in the cell. It produces ATP and it uses oxygen. So it's what the oxygen is for inside the cell. You know, those kinds of things. Okay? This isn't going to come up just in this unit, because we're going to refer back to this in other units as well. We're going to talk about it in the animal systems unit, because we're going to talk about how cellular function is related to systems function. So this is sort of just general information about a cell that if you didn't lock into your brain in grade 10, it's one of those like basic biology things that you're pretty much going to draw upon no matter where you go in bio. So this is, you know, kind of core information for biology that you need to know. So if you haven't stored it away yet, you may consider doing that. Again, for me, I would make flashcards. I would have a picture on the front with name on the, or maybe just the picture on the front, name on the top of the back, and function below it. You can cover up various parts, flip them over, learn how to name them. Again, it's up to you, but I would strongly consider adding these to your memory banks here. Especially the way we're doing the course this year, which is with open book quizzes, it's not essential in this course that you do a ton of memorization, especially names of things. I mean, you could potentially look this up while you're writing the quiz, right? It'll take you time, so it'll make it harder. But you don't necessarily have to memorize. That being said, that is purely a product of the way that we're doing the course this year. Normally, I would ask you to memorize these. And certainly in subsequent courses, people are going to assume that you have. So just because I'm not testing you on whether or not you can recall what all the different parts are called, it doesn't mean that you probably shouldn't know that at this point. So, or maybe not at this point, but at least after a review. Okay, so I'll put that out to you. It's nothing you need to stress over, but just consider that a lot of like the core biology information that you learn in this course, you need to know that for your future biology career. So there's a little bit of review at the beginning of university too. You know, you take an intro course to cell biology. Yeah, sure, they're going to go over the organelles again, but they're going to do it very briefly, and then they're going to expect that you definitely know what they all do at that point. So it's usually better to go in with the basic knowledge already. Okay, there are some questions that are afterwards here. This is a test your own knowledge homework question portion where you just go through and see if you can fill in the blanks. It is directly calling on the information from the descriptions above here, okay? So go through them first, see what you can remember, and then you can use the above part here to help you answer the questions, but the idea here is that it's helping you with recall, okay? It's testing the information you've just reviewed, trying to get you to recall it, to synthesize, to think about that information in order to sort of better cement things into your memory. Okay, so that you're going to complete that right after that. I'm very briefly now, this should take me maybe 5 to 7 minutes, going to talk a little bit about DNA, just the basic structure. And then tomorrow, we're going to get into, actually, you know what? Hold on. I want to leave you time to work on this stuff too, so I don't want to like really hit you over the head. Where do we start on day 6? Protein synthesis. That's a busy day. Okay. Okay, I'm gonna just start this, but I think that I'll finish it tomorrow because I do want to give you some time. Oh no, you got to write your quiz too. Never mind. I'm going to start this tomorrow, okay? I realize in Learning Block 3, people want some time to start their quiz as well. So I'll let you work on this. So it's just up to the end of the questions here, checking your understanding of the organelles. Okay, let's just leave it right there. And then the DNA stuff we'll get into tomorrow morning. Any questions about this stuff? I mean, this is review from last year, but... And I've also, if you're not sure what something is after looking it up, I've posted this, so you're welcome to look at the completed note as well. But the idea is that you're, attempting to recall stuff from last year. Okay, I don't see any questions. Remember that you should maybe take a shot at the quiz today. If you're really not ready, it's not a big deal. Tomorrow during Learning Block 3 would be your next ideal time. That's really all I have to say. I'm going to give you time to work on this. Okay.